Hi, this is Miss Litton, and this is a teeny tiny review with how many of you? 16? 16 biological bad mamma jammas here today. Excellent. First thing we want to talk about is make sure you know the 3 parts cell theory. Okay, cell theory. So all living things are composed of cells. Cells are the structural and functional unit of all organisms, and cells only come from pre-existing cells. So those are the 3 parts of the cell theory. I could ask you a question related to that. The other thing we need to remember is that cells need to be what? Small. Why, Appers, you ought to be able to answer this. Why must cells be small? You need a what? Large surface area per volume. Who has, well, first of all, what kind of cells are these? Eukaryotic or prokaryotic? Eukaryotic. Eukaryotic, and you can tell because they have membrane enclosed organelles inside, yes? Okay. So do they have a larger surface area to volume ratio, or do prokaryotic cells have a larger surface area to volume? Prokaryotic. Why? Because they are smaller. The smaller the cell, the larger the surface area per volume. All right, next. And we had some little notes about that, which you can go back and take a look at. Then we focused just on prokaryotic cells. So we looked at them from the outside to the inside and then everything that is inside. So from the outside, we talked about that there was some sort of capsule here, and they had these little fimbriae. Do you remember what the fimbriae's job was?
To latch on to things.
Yeah, to latch on things to attach. Then we move inside, and they would have a cell wall. And then you go in a little bit deeper, and they would have a cell membrane. Do all cells have a cell membrane?
No. Only plants.
Yes. Yes. All cells have cell membranes. Okay. Some cells have cell walls, right? Okay. But they have a cell membrane, and remember, what's the job of the cell membrane?
To allow stuff in and out of the cell.
In and out. Now we have already started in chapter 4 talking about how things can cross a cell membrane, right? What are the ways things can cross a cell membrane? It can go through the phospholipid bilayer, but not if you're large or charged. You can use a channel or a carrier, and you can do endocytosis and exocytosis. Those are all the ways. So we know why it's critical that you're not a cell unless you have a cell membrane, right? All right, so that's its job, and now you remember you can see the phospholipid bilayer, hydrophobic region in the middle, hydrophilic, and then these are proteins, yes? So we're spending a lot of time in chapter 4 just on that. Okay, going inside again, I see this meso blue right here. What is this meso blue right there? Well, there is the cytoplasm, which is this whole area, but I'm talking about this region, which you cannot see because I highlighted it in green. This region right here, nucleoid. Now, it is not a nucleus because it doesn't have a nuclear envelope around it. It's just all of its DNA just in this one region. Okay, that's the nucleoid region. What are all these little dots in here? Ribosomes. What happens on a ribosome?
It's the workbench for protein synthesis.
Protein synthesis. What is this thing right here? Flagella. What does the flagella do? Swim movement. Okay. Then there is this pili right here, which looks like exactly like the fimbriae in this picture. What do the pili do?
The
sexual pili. So you can use and you can develop these pili with multiple different cells, and you exchange DNA across it. This gives them a chance at maybe getting some new genes in the exchange that make them better able to survive in a changing environment. All right, and here is another picture where we went all over all the little different topics. You can see the sex pilus up at the top. Maybe. Fimbriae, tell me again what those are for?
To latch on to things.
Latch on to things. Flagellum is for movement. Ribosomes, tell me again. Protein synthesis. Nucleoid, what's in the nucleoid region? DNA. And plasma membrane is where all exchange comes across. Cell wall, I didn't have you memorize this, but peptidoglycan, uppers, you will. Okay, and then the glycocalyx are the polysaccharides. Now, did we talk about glycocalyx when we talked about the cell membrane? Remember the glycolipids and the glycoproteins? We just learned that, okay, when we focused on the cell membrane. So that's carbohydrates. Now, also on this page, we have bacteria and I have archaea. Why do I have a picture of archaea on the same page as I have a picture of bacteria?
Because they're both prokaryotes.
Boom. They're both prokaryotes. Good job, you guys. Both prokaryotes. Those are two different domains, right? Bacteria and archaea. The eukaryotic cells are in the domain eukarya. Hence the name eukaryotes, right? All right. Good. Ask the person next to you. How much of that, what do you understand? What do you remember? Are you feeling good about that? Something you need to focus on?
I'm pretty solid with that.
I'm not going to lie. I'm solid. Okay. Now, is this chromosome the only DNA this bacteria might have? No. You could have another one. What's that called?
It's the nucleolus, right?
Ooh, okay. Let's think what that is. You're wrong, but that's okay because you were trying. Remember, the nucleolus is inside the nucleus of a eukaryotic cell, and that's where our RNA is made. Okay? There's a different name. I'm looking at my appers now. What is the extra bonus DNA? What's it called? Plasmids. Yes. Remember plasmids? The extra ring of DNA? Oh, whoa, I'm flying. Look at that. All right. Watch it fly. Whoa. Okay, sorry. It's Friday. I'm tired. I think I say that every day. Plasmids are an extra ring of DNA. Remember I told you we're going to genetically engineer those? Yes? Little extra pieces of DNA. Is this how the bacterial chromosome would actually look? No, it would be a mess, like something out of your hairbrush. Good. And if I was going to pick a poster child for bacteria, who would I pick? Cyanobacteria. Because they do nitrogen fixation, and they probably did what you were starting to say, oxygen revolution. Yeah, so that's a very important bacteria. Okay, and like I mentioned before, these are your three domains, eukarya, archaea, and bacteria. Archaea and bacteria are both prokaryotic cells. Archaea are different in that their membranes are different, their cell walls are different, and what else is different about them?
They live in extreme environments.
They live in extreme environments, exactly. Okay, eukaryotic cells right here, okay, probably evolved from prokaryotic cells. And we talked about the endosymbiotic hypothesis, and we focused on that once we started talking about mitochondria and chloroplast. So I'll jump back into that in just a little bit. Okay, so let's jump into eukaryotic cells. I would, like I suggested, and you probably have them in your group shared notes, is to have a labeled and unlabeled picture of a plant cell and a labeled and unlabeled picture of an animal cell. So that you can test yourself and make sure you know all the organelles and you know what they do. Hi, they came by and there weren't any appers who needed help.
Okay.
Yeah, unless any appers in the room right now want free tutoring. They're busy on their lab. Thank you, sweetheart.
No problem.
Okay. So, and honor students, you can get free tutoring too. These are people, I mean, because if you paid for a tutor, it's very, very expensive. But these are kids who passed the AP Bio exam, so they definitely know all of your material as well. And they come on on days I normally give you reviews on 1, 3, 5 days. But if it's an open period, if you come here, they hang out at Lab Station 5 on 1, 3, 5 day. Yeah, wait. 1, 3, 5 days. Yeah. 1, 3, 5 days at Lab Station 5, and they are free tutoring if you ever needed it. Okay, so make sure you can recognize all the different structures. We'll just hit the big ticket items. We'll kind of run through. And when I say nucleus, try to come up with as many things as you can think about that we talked about the nucleus. Just start jotting them down in your group shared notes. Be like, boom, boom, that's such a pretty ring. Just start typing some things you remember about the nucleus. See what you can remember. Okay, did you say that it contains the chromosomes?
Yes.
Did you say that the chromosomes were linear?
No.
Okay, because the bacterial chromosomes are what? I'm drawing it.
Circle.
Circular, yeah. But eukaryotic chromosomes are linear. Did you say that they are also associated with proteins? When we talked about how to organize them. Did you say that there's a nucleolus inside the nucleus? This is that dark region right here, and that's where our RNA is made. Okay, when I say the DNA is associated with proteins, that's because when we wind up our DNA to form a chromosome, which only happens during cell division, the DNA winds itself on these histone proteins. 8 histones form a nucleosome. You do not need to memorize that. AP, you do. But the DNA winds up on these proteins, and it helps organize it. This would be a perfect example. Okay, so her hair is all organized. So these would be like sister chromatids that are going to separate during the anaphase of mitosis. Your DNA would all wind up. To make sure that you get, see your 2 ponytails? It would be like these 2 ponytails right here, so that each cell gets all the DNA that it needs, okay, in order to survive. Here's another picture of that, the DNA winding up, and you can see these chromosomes are linear as opposed to the circular chromosome that you have in bacterium that never appears like that, and instead it's all scrunched up and appears like this. All right, so then next topic I want you to think about is ER. Right, everything you know about ER right now, start jotting things down. See what you know about ER. Are there types of ER? What do they do? See what you can remember. You can do this at home too. Oh, I'm showing things. Whoops. Don't look. What are the 2 types of ER? What gets manufactured on them, through them? Okay, so tell me, what are the 2 types of ER?
Smooth and smooth.
Rough and smooth, good. The rough one is closer to the nucleus. What makes the rough ER rough?
Has
Ribosomes. And then on ribosomes, you already told me it's in charge of protein synthesis, but I want you to remember that there is a small and a large ribosomal subunit. And messenger RNA is read on the ribosome, and you use that information to build a protein. Okay? Now, if I ask you where protein synthesis occurs, you're not going to tell me the rough ER. What are you going to tell me?
Ribosomes.
Ribosomes. It just so happens that some ribosomes are on the ER and some are out in the cytosol, out in the fluid. Okay? Once this protein is made, though, it can move to the interior of the rough ER and move through the rough ER, okay, and be packaged and sent where? Where does it go after it goes to the ER? Golgi body, and another name for that is Golgi apparatus. Lipids are made in the smooth ER, okay, lipids are made in the smooth ER, but proteins are made on the ribosome and then they move into the rough ER. And that's your next transition. If you get your bearings here in the bottom left, the protein is in green here inside the rough ER. I'm pointing right here, but you can't see me. Right there, you can see the green right there. And then it's going to be put in a vesicle and sent over to the Golgi apparatus. Lipids are going to be made in the smooth ER and sent over to the Golgi apparatus. Now, write down everything you can remember about Golgi apparatus or Golgi bodies. You can do that at home too while you're waiting. Don't shake your head at me. Actually try. Now you're rolling your eyes at home. I know what you're doing. You shouldn't have waited until late Sunday night to listen to this video. Who should we shout out to that should be here today that's not? What? Shout out their names. Say, are you watching? Ask them. Of course, every time. It's like the only name they can say.
Be here. I hope you're listening to this.
Yes, I hope you're listening to
You
this.
told me that you'd listen to it, so.
All right, here we go. You're just throwing them right underneath the bus and rolling over. Okay. For Golgi apparatus, if I was going to pick a job, What would be the job? I would describe the
UPS man.
Yes.
Processes, packages, and delivers, okay? So whatever it is, it packages. The cis side faces the nucleus, trans side faces away. It packages. Maybe it's getting packaged for this. What was it? Exocytosis, some sort of secretion. Or maybe it's packaging something to stay within the cell. Now, speaking of things that might stay within the cell, okay, we could talk about a lysosome. Write down what you know about a lysosome. Don't say it out loud, just type it. See if you come up with it.
Wait, I have a question.
Yes,
Are the lipids packaged instead or the proteins?
both will move. Everything will move to the Golgi. From that ER, whether rough or smooth, it'll move to the Golgi. Golgi puts the address label on it where it's supposed to go. Inside the cell, outside the cell. Okay, give me one word for lysosome. Stomach. It is the stomach of the cell. Yeah. It digests things that were brought in by endocytosis. Things will be absorbed out of it, and then it'll go to the edge of the cell membrane and do what? Exocytosis to get rid of it. Okay? How's your data back there? Is it okay? What minute are you at?
19.
19, and how many have risen?
4, 12, 12.
Oh, really? 4 rows in your 0%?
Yeah.
How about you? Did you guys get it in your 0%?
7, 6, and 9.
Okay, so that's really interesting. So I'll let you know, when I was restocking the solutions at lunch, a thought went through my head going, did I put 1% in the 0% container? So now I know I did. That's awesome. It doesn't affect you. It's fine. I thought, I wonder if I should pour it all out and re-put it in there, but I thought, I'll just add a little bit more. That's good to know. Either that or they were really photosynthetic. Did you see a big difference? Tell me your numbers again. What'd you get? When was your ET50?
We're still on minute 17.
But how many do you have up? Like, do you have at least half of them up? 5 up?
Yeah.
So when did you get 5 up? Six, 6.5, and 12?
6.5.
But the other ones were 12? So you got 5 up in the 0%
I need to know.
at 6 minutes? And not till 12 minutes in the 3%? Now that's weird. I didn't make that mistake. What about you?
For 1%, we got 5 up at 5 minutes, and in the 2%, we had 6 up at 6 minutes.
Okay, and how about 0%?
0, we're just at 4.
Oh, so they haven't even been 5. Okay, that's good. Okay, all right. Now, okay, okay, so let's go back here. So here, this is a great review picture that you have in your book. So sorry for that, I just need to check on AP. So you can see the nucleus, you can see the smooth ER and the rough ER both delivering to the Golgi, and then the Golgi will have tagged it either for exocytosis or to remain within the cell. Okay, then we also talked about, we talked about peroxisomes. Okay, so we talked about, peroxisomes, and peroxisomes contain an enzyme inside of them. What do they contain inside of them? Starts with a C. Catalase. Remember when I talked to you about catalase, and it breaks down hydrogen peroxide into what? Oxygen and water. You didn't remember that one at all. So that's a peroxisome. How are you going to remember that? Hydrogen peroxide. Yeah. So it breaks down. Hydrogen peroxide is a metabolic byproduct, and your cells need to break it down. And so peroxisomes contain an enzyme called catalase that breaks down hydrogen peroxide into oxygen and water. You're going to remember oxygen because when you pour hydrogen peroxide on a cup, what does it do? It bubbles. That's the oxygen. Okay? All right. Then we started talking about vacuoles, and what kind of cells have the largest vacuoles? Plants, because they use it to create what?
Structure.
Well, yeah, structure, trigger pressure, and it pushes against the wall so that the plant stays upright. You can have smaller, I kind of think of it as a multi-purpose organelle, And you can have smaller ones that are located in animal cells or contractile vacuoles that secrete water out of protists. Then we talked about the two energy organelles, and we started talking about the endosymbiotic hypothesis. And your idea there was this was a good place for us to insert how a eukaryotic cell could have evolved from a prokaryotic cell. And the reason is both mitochondria and chloroplasts have all the appearances of a bacterial cell. One that does photosynthesis and one that does cellular respiration. Can you remember some of the evidence about chloroplasts and mitochondria that makes you think that they were once free living? And jot those down on your little notes you're taking right now. Mitochondria and chloroplasts have their own what? DNA, and it's in the shape of a circle, just like bacteria. They have their own membranes. Well, they have double membranes, which is like they got engulfed, and they have their own, starts with an R. Ribosomes to do their own protein synthesis, and they are self-replicating. They can make more of themselves. So mitochondria and chloroplasts are like a free-living bacterial cell that went to live in another bacterial cell. So that's how you could have gotten those organelles. How could you get ER, or how could you get a nucleus? By folding membranes. Yeah, membranes folding in. Exactly. Okay. Then we talked about that chloroplasts are the site of photosynthesis. We talked about mitochondria are the site of cellular respiration. The last thing we talked about was the cytoskeleton. Like you have a skeleton for support, so do cells. We said that there were 3 different types. There was actin. I always think active actin because it's small. It's very active. That actin is the tiniest one. Then the intermediate filaments are intermediate, medium, and the microtubules are the biggest ones in diameter. These 3 cytoskeletons work together. Anytime a cell membrane is doing this or this, endo or exocytosis, any kind of movement, when organelles need to move within the cell, your cytoskeleton is involved. And remember how we looked at Walking Man? Okay? And then we said you could use microtubules, 9 triplets of microtubules, the largest cytoskeleton, to form centrioles, which look like what to us? Churros. And animal cells have them, but plant cells don't. Yes? Oh, 9 triplets. So 1, 2, there's 9 of them? Yeah, 9 triplets of microtubules to form centrioles. Okay? So animal cells have them. Plant cells do not. And they do cell division fine, but you get 2 sets of them when a cell is dividing. The other place you see this structure is the thing that anchors cilia and flagella is also 9 groups of 3, and they're called basal bodies, and they anchor cilia and flagella. Cilia and flagella are slightly different though. If you look at them, they are not 9 groups of 3, they are 9 groups of 2. And then they have 2 singles in the middle. And if you looked at them with an electron microscope, here you have 9 groups of 2. So that would be cilia and flagella if I showed you a picture like that. If I showed you 9 groups of 3, you would know those were centrioles or basal bodies. Exactly. Okay? Basal bodies anchor cilia and flagella.
Oh, and then the cilia and flagella are the ones that are 9 groups of 2.
Yes. Mm-hmm. And then this is just like structures, that we had. If you want to, I can start. This is, I think it has 15 questions. It's a review, if you would like to do those. The only thing it doesn't have is I didn't put any of the pictures in here. So go in as something you're looking forward to this weekend, I guess, since the weekend's about to start. Of course, T-Bone. Just going to get the other name out. Okay, there you go. And if you're at home, this would be a great time for you to get a piece of paper real quick while I'm talking to you and number from 1 to 15 so that you can answer the questions as we go along. Here we go.
She's gonna.
Leave it. Yeah.
Capitalize the X.
No, don't worry about it. You do it all next class. Yeah. I'm curious to see everybody's data, to see if I just wrote it. And I'm like, did I?
Yeah, because
Did we get it started on the bottom?
it is.
It determines what is going in.
Oh, is it too? Okay, all right. Okay, so you're committing at home as well. Here's question number 2. And question number 3. When you guys are answering these questions and choosing an organelle, imagine what it looks like. So you reaffirm what it looks like. Okay, right now in your brain, because we just went over everything.
What color are they, like in real life?
Color doesn't matter. Nothing. You wouldn't even see them. They're pretty clear. The only reason they're colored is because somebody stained them so you could see them. Yeah. Except for chloroplasts, and those are green because they do photosynthesis. Yeah. Here's question 5. Here's question 6. You can do it. Come on, commit to an answer. Here's question 7. Here's question 8. And here's question 9.
Is asking how do I pronounce this, and it's really simple.
And 10.
I'm just waiting.
Bye, sweethearts. Thanks, guys. Have good weekends. And here's 11. And number 12.
No, it's century.
And number 13.
Central. No, yes it is.
We have a couple extra names in here.
T-Bone's house, yes, sir.
Okay, and I'm going to use this moment to remind you to do your,
Highly suggested.
yes, your additional objectives. Make sure you do those. And you're going to study the diagrams too, yeah? I think we have one. Okay, sleep needs to just answer 2 more questions. You got it, sleep. Come on, sleep. You got it. And soccer, 1 more question. These other children are gone. 13. Okay, here we go. Okay, organelle is a powerhouse, mitochondria. Everybody got it? Who decides who goes in and out? Cell membrane. Uh-oh, not the cell wall. Cell wall, water and gases can diffuse through it, but it doesn't decide what goes in and what goes out. Number 3, processes, packages, and distributes. Give me a job description. That would be the what man? UPS man, Golgi apparatus. Number 4, everybody got it? Lysosome is the stomach. Number 5, permeable to gases, supports, protects. That's your cell wall. Not a chloroplast. Number 6, control center is the nucleus. 7, ER, almost everybody got that. Number 8, converts light energy into carbohydrates, chloroplasts. Multi-purpose is vacuoles. Somebody picked lysosome. What would I pick for lysosome? Stomach, digest. Good. Number 10, cytoprotein synthesis is ribosome. Remember, I specifically said don't put ER, but we still have somebody doing that. Okay? Definitely the cell membrane doesn't. Which of the structures will be found in both plant and animal cells? Mitochondria, not centrioles. Centrioles are only found in animal cells. And yeah, not cell walls. You're an animal cell. You don't have cell walls. Okay. Number 12, 9 sets of 3, centrioles and basal bodies. 9 sets of 3. Cilium flagella have 9 sets of 2. Okay, and that was question 13. Do you have any questions you want to ask me? Anything you're confused about?
What do centrioles do?
No, I don't know. Nobody knows. They're found in animal cells, and they sit in what's called a centrosome, or a microtubule organizing center. And microtubules shoot out and help the cell to divide. But plant cells divide just fine without them. Yeah, the churros, yeah.
So those exist?
Yes? I don't know. I haven't written it yet. Yeah. As soon as I write it, I'll tell you. Okay? Have good weekends. Make good choices. Remember, I love you.